Chapter 3 Review, Cell Biology. In this lecture, we need to review just enough cell biology to get us through the A and P series. We'll cover a number of organelles that are important to anatomy and skip those that are not. First off, most cells cannot be seen under the microscope on their own. We must use chemicals that stain parts of them a darker color. The most common stains that you will be looking at are the hematoxylin and eosin stains, which make some parts of the cell pink and others purple. One of the first demonstrations in a cell biology class is to look at the letter E cut from a newspaper under the microscope. Both printed newspapers and microscopes are becoming rarer these days, so let's just go through this theoretically. If I were to tell you the size of that letter E was 2 millimeters square, and that the average cell is roughly 10 micrometers in diameter, you should be able to calculate the number of cells that would fit within that letter E. First off, you would remember that 2 millimeters is equal to 2,000 micrometers. And therefore, if we divide that by 10 micrometers, you should be able to fit about 200 cells across that letter E. We multiply that by another 200 upwards, and we get about 40,000 cells. Of course, that letter E is not a square. Let's say it's only roughly a quarter full. We could get about 10,000 cells or so into the letter E. So those are pretty small. Moving on, our next concept is the difference between hydrophilic and hydrophobic. Hydrophilic molecules were those that could form hydrogen bonds with water, and hydrophobic could not. Therefore, if we dump a hydrophobic molecule into water, that'll be interfering with some hydrogen bonds. It'll interfere with the least number of hydrogen bonds if all of the hydrophobic molecules glom together. And this is what we see. These two substances would not mix. Instead, I'd have regions of hydrophobic molecules and regions of hydrophilic molecules. Phospholipids can do this in a special way. Remember that a phospholipid has this hydrophilic head group and the two hydrophobic tails. Therefore, if I dump a bunch of phospholipids into an aqueous solution, they will orient themselves so that the head groups are facing the water, whereas the hydrophobic tails face one another. And this would form a lipid bilayer. This phospholipid bilayer is the basis for the plasma membrane of cells. The phospholipids form a barrier to water-soluble molecules. They cannot get across the lipid bilayer because water-soluble molecules will not be able to penetrate the hydrophobic core. To get molecules like glucose or ions like sodium into the cell will require transmembrane proteins that can act as transporters. These proteins will be able to bind and move hydrophilic molecules across the plasma membrane. We therefore say that the plasma membrane is semi-permeable. It is permeable to some substances, but not to others. These molecules are not crystallized, though. They can move around from side to side. What they can't do is flip around or leave the membrane, but they can diffuse from side to side. We call this the fluid mosaic model. Our plasma membrane is a mosaic of phospholipids and proteins that can move around sideways. They do not reverse themselves or exit, however. Next up, we need to cover a number of cell-to-cell and cell-to-protein connections. The three major cell-to-cell connections are tight junctions, anchoring junctions, and gap junctions, whereas the cell-to-extracellular matrix connection are the integrins. First off, tight junctions completely encircle the cell, forming a water-impermeable barrier between cells. This will differentiate the apical surface of the cell from the basolateral sides. Because this is water impermeable, solutes in water will not be able to diffuse between cells. So if we look at a bunch of cells here from the side view, I may have transport proteins in the plasma membrane capable of pumping glucose into the cell. On the other surface, I may have proteins that pump glucose out of the cell. And if this is my intestines, I would then pump glucose in one direction, from my food into the blood. And if I spend a lot of energy pumping all of these nutrients into my bloodstream, I would not want them diffusing down their concentration gradient. That is what they would do if there were any gaps between these cells. Tight junctions ensure that there are no gaps. Otherwise, both nutrients and blood could leak back into the lumen of my intestines and wind up in my feces. Furthermore, because of the fluid mosaic model, these proteins could also diffuse. Let's look at just these proteins, though. I have proteins that pump glucose into the cell on the apical surface, and the ones that pump it out on the basolateral surface. And there are concentration gradients for both of these proteins. If they were allowed to diffuse down their concentration gradient, staying within the plasma membrane but sliding through it, I would wind up with an equal distribution of these proteins on both surfaces. This is the type of thing that tight junctions prevent. In this sort of a situation, glucose would be pumped into the cell and then right back out, not going in the direction that I want, which would be strictly from the lumen of my intestines into the bloodstream. Next up are the anchoring junctions. These are not water impermeable. These junctions are strong. They anchor the cytoskeleton of one cell to the next cell to the next cell. Working as a unit, these cells are now very strong all together. Lastly are the gap junctions. These can form a pore between cells, which will allow communication from one cell to the next. These will be very important when we discuss the heart, where electrical signals will pass from one heart muscle cell to the next one. Cells are sometimes anchored not to other cells, but to the extracellular matrix, or proteins and glycoproteins found outside of cells. These anchors are called the integrins. They can help cells to move through this matrix, or to stay anchored to the matrix, or to even just get information about where they are located. Next up, we need to review a little bit more about equilibrium. Equilibrium was the passive movement of molecules from a region of high concentration to low concentration. Osmosis was the movement of water from high concentration to low concentration as well. However, we usually say from low solute to high solute. That means where water is the highest in concentration, we've got the lowest amount of solutes. Conversely, where water is the lowest in concentration, that means there's the highest amount of solutes. So equilibrium was the ability to reach a new steady state passively. In the winter, if I were to open up all of my windows, the temperature of my house would eventually reach equilibrium with the outside environment. My house temperature would reach a new steady state. Equilibrium is a completely passive process that happens because of the random motion of molecules. If these molecules here move randomly, they will ultimately wind up half on one side of our border and half on the other side of the border. At this point, they have reached equilibrium. If they were to continue to move, I would still expect there to be roughly 3 on each side of the border, but it may not be the exact same 3. We call that dynamic equilibrium. So molecules can do this as long as they are able to move. These hydrophobic molecules would eventually reach equilibrium, meaning there would be an equal concentration outside of the cell as inside of the cell. Those green molecules do not care about the equilibrium of the yellow molecules. If I added a bunch of yellow molecules, that would not change the equilibrium of the green ones, even if those yellow ones were not able to enter the cell. If they did, eventually both of these would reach equilibrium. And of course, adding a third type of molecule would not change the equilibrium of the first two. Equilibrium can become more complicated when we discuss cells. Let's say I've got 2 chambers of liquid with a solute in one of them, and a semi-permeable membrane that allows that solute to move. The solute would move from the region of high concentration to low concentration until we reached equilibrium. If I added a second solute that was also capable of moving across that membrane, both solutes would eventually reach equilibrium. Here's where things get more complicated. Let's say I have an enzyme in the left-hand chamber that can convert the big purple solute into a white one that is not capable of moving across the membrane. What would happen next? I would now have a new concentration gradient. Purple would be highly concentrated on one side and white on the other, but the white one cannot diffuse down its concentration gradient. The purple one still can, so purple molecules will move over to the left side of the chamber, and as they do, they would get converted into white ones because of this enzyme. So I would maintain this concentration gradient until all of the purple molecules had moved over to the left side of the chamber and been converted into the white ones. Osmosis is the name for the diffusion of water. And again, we say it moves from a region of low solute to high solute concentration. So if I had a bunch of solutes in the left-hand chamber and only water could move across the membrane, water would move from right to left towards the region of high solute concentration. The left-hand chamber would begin to fill up. We still have a low to high solute concentration, however. Not all of the water will move. And that is because there are more forces than just osmosis. Eventually, the force acting down due to gravity will balance the force driving water into the left-hand chamber. If I were to add an equal number of molecules to the right-hand chamber, I would now have a new osmotic gradient. The left-hand side would have a lower concentration of solutes. Even though it has the same number of solutes, they are in a larger volume, hence that is a lower concentration, and water would move back to the right-hand side until we reached equilibrium, or an equal volume of water and an equal number of solutes on both sides. It wouldn't matter which solutes those were. I could change these molecules. I would still be at equilibrium as long as the concentration of solutes is equal. So that's the plasma membrane, and diffusion and osmosis, which can occur across the membrane, presuming I have the right type of proteins. Next up, let's discuss endocytosis and exocytosis. The plasma membrane can change its shape to engulf materials from outside of the cell, or do the exact opposite. I can take vesicles and fuse them to the plasma membrane to remove molecules from the cell. First off, we have to consider that what is found inside of a vesicle, even though that vesicle is inside of the cell, the lumen or the hollow center of that vesicle, is considered to be equal to the outside of the cell. The same way that if a bunch of raisins were in the center of a bagel, we would not consider that a raisin bagel. It would be a bunch of raisins in the bagel hole. To actually get raisins into the bagel, they would need to be in the bagel itself, not in the bagel hole. Similarly, if we were to push on the plasma membrane and force it to invaginate inwards until it pinched off to form a vesicle, the lumen of that vesicle is the equivalent of the outside of the cell. The plasma membrane is the phospholipid bilayer surrounding the cell. The membrane surrounding the vesicle is the equivalent. It's still a phospholipid bilayer, but we don't call it the plasma membrane because it's not at the surface. But it could easily join up with the plasma membrane and become a part of it once again. If it were to do so, anything that was inside of that vesicle would now be outside of the cell. That's why we consider those two spaces to be equivalent. To truly be inside of the cell, something has to be in the cytoplasm. So the cytoplasm contains water, electrolytes, and other solutes, and all of the organelles. This makes cytoplasm very similar to fruit-filled jello. The nucleus is a large organelle in the cell that contains almost all of the DNA of the cell. This DNA can be converted into RNA, and ribosomes can also be made in the nucleus and exported outside. You may remember looking at chromosomes in your cell biology class. We're not going to see them frequently in this class because mitosis is a fairly uncommon event. So most of the DNA is just going to be sitting there stringy in the nucleus. Not all of it, however, and that's because not all cells need all of their genes found in the nucleus. For instance, an epithelial cell is never going to turn on a bunch of genes that a muscle cell would transcribe and translate in order to cause muscle contractions. The epithelial cell, therefore, can pack these genes away because they're not needed. This packing is in the form of DNA methylation and then packaging around histone proteins. So that's the cytoplasm and the nucleus. Next up, we've got a bunch of organelles to cover. The smooth endoplasmic reticulum is a series of interconnected tubes within the cytoplasm. These tubes can synthesize different lipids, they can detoxify certain toxins or poisons, and it can also store calcium. The rough endoplasmic reticulum is the region of these tubes that appears rough under the microscope because it is covered in ribosomes. These ribosomes synthesize proteins that will either be secreted from the cell or embedded within the plasma membrane. Here are two pictures of a B cell at two different times in its lifespan. On the left is the naive B cell, and on the right is when the B cell is activated. You can see a huge increase in the amount of rough endoplasmic reticulum, and that's because this activated B cell has just one job, which is to secrete proteins called antibodies. There are also ribosomes just floating in the cytoplasm. These will synthesize proteins that remain within the cell. So that's the endoplasmic reticulum and ribosomes. Next up, a protein that is made on the rough endoplasmic reticulum will travel next to the Golgi apparatus, where that protein can be modified and then shipped to the surface of the cell. So if I have a ribosome bound to a piece of mRNA, and it synthesizes a protein inside of the rough endoplasmic reticulum. This protein will next go to the Golgi and then ultimately outside of the cell. Membrane proteins are also synthesized on the rough endoplasmic reticulum, but they do not go completely through. They stay embedded in the phospholipid bilayer. Thus, when this vesicle is ultimately fused with the plasma membrane, this protein becomes a part of the plasma membrane. So our big summary is that most of the DNA is in the nucleus. It can be transcribed into a messenger RNA, which can be translated by ribosomes. If these ribosomes dock on the rough endoplasmic reticulum, these proteins will next travel to the Golgi apparatus via a vesicle. They can be modified in the Golgi and then travel via another vesicle to be secreted from the cell. Next up are lysosomes. These are small vesicles full of acids and digestive enzymes that are capable of destroying materials that have been endocytosed by the cell or digesting and recycling old organelles. Tay-Sachs disease is an example of a disease involving lysosomes. It's an inherited disorder where one particular type of lipid cannot be metabolized. And so it builds up within specific cells in the brain and spinal cord, leading to damage of the central nervous system. Next up are vacuoles. In your cell biology prerequisite, you probably learned about the function of vacuoles in plant cells, which were to store nutrients and regulate water pressure. Human cells really don't have vacuoles. Lysosomes are the tiny vacuoles that human cells find. There's one exception. We can find a vacuole inside of adipose tissue. Adipocytes will store a lot of triglycerides inside of a large central vacuole. This vacuole becomes so large it pushes the nucleus off to the edge of the cell. And this is what those cells would look like under the microscope. It's mostly vacuole, but we can see little nucleuses here as well. There we are. There's a nucleus. There's another one over there. So that's the Golgi apparatus, lysosomes, and the one example of a vacuole that we can find in the human body. Next up, the mitochondria are responsible for generating most of the ATP inside of a human cell. They do this using oxygen to break down glucose and harness some of that energy in the form of ATP. Next up, the cytoskeleton is not really an organelle, but a network of proteins inside of the cytoplasm. There are three major types of cytoskeletal fibers: actin, microtubules, and the intermediate filaments. These fibers give the cells their shape and allow them to change their shape, for instance, when they move. Actin is made of small globular proteins that can stick to one another to form a small fiber. These are very important in cell motion and shape change. The microtubules are bigger and stronger. These give the cell rigidity and shape. Organelles can also move along microtubules as if they were little freeways throughout the cell. The intermediate filaments are intermediate in size, and these also help give the cell their shape. Microtubules are also the important factors that make up cilia and flagella, structures on the surface of the cell that allow them to move. Cilia in the human body move substances across the cells, like mucus. The only flagella that we will see is on a sperm cell, and that gives those cells motility. Microvilli, which are very difficult to see under our microscopes and really just appear as a fuzzy border seen to the right, are made of actin. Microvilli do not move, but they are small finger-like projections that greatly increase the surface area of a cell. This is important for cells that need to absorb nutrients, for instance, in the digestive system. So to summarize, if one cell was roughly the size of a football field, the nucleus would be pretty big, about 50 feet across, and the mitochondria would also be fairly large, about 18 feet across. Lysosomes, on the other hand, would be pretty small, maybe just inches, as would ribosomes and microtubules and other filaments of the cytoskeleton. They are tiny, but because they are numerous, can be very strong. The last topic for this lecture is mitosis, or cell division. First, a cell must decide when it's going to go through mitosis. It'll make these decisions at different cell cycle checkpoints, times when the cell has to decide whether it will move forward or halt its progression through the cell cycle. The cell may look towards environmental factors to make this decision, such as whether the right hormone is present or whether cell density is too low or too high. When the genes that regulate cell cycle checkpoints become mutated, this can lead to cancer or uncontrolled cell division. When a cell undergoes mitosis, it must first duplicate its DNA during the S phase. The DNA is copied by semi-conservative DNA replication. The two strands of DNA are separated and copied by an enzyme called DNA polymerase. The DNA methylation that we discussed earlier is also copied. Therefore, an epithelial cell that has turned off its muscle contraction proteins, when it duplicates its DNA, the muscle contraction proteins will still be inhibited and stored in histones. Therefore, epithelial cells give rise only to epithelial cells when they undergo mitosis. They cannot produce muscle cells. Conversely, muscle cells can only produce other muscle cells, not epithelial cells. To produce different cell types requires the activity of a stem cell, a cell that has not decided what it's going to become when it grows up and has not methylated a significant portion of its genes. So that's the mitochondria, the cytoskeleton, and mitosis, and that should be just enough cell biology to get us through the anatomy and physiology series.